The hypothalamus is small, old, and gnarled, and what it does ranges from the mundane to the sublime. It determines how tall we will grow, how fat we will be, whether we will freeze, fight, or flee. The rhythms of days and of seasons are beaten by its drums. Led by the hypothalamus, we preen and woo, and the sap rises in our loins. A man produces sperm, and in a woman, the ovarian cycle turns. Different regions of the hypothalamus control gluttony, anger, sloth, obesity, drinking, addiction, stress, love, and hate, and sex. All human life is there. Welcome to Leafing Through Science, where we dive into the pages of popular books in the life sciences with the writers who made them happen, from new releases to timeless classics. I'm your host, Hedi Young, and today we're venturing into a part of the brain that most of us have never heard of, and yet it might be pulling more strings than we realize. As human beings, we like to think of ourselves as reasonable, but how much of what we do is really governed by reason? How often are the reasons we give merely self-serving narratives? And how much is actually driven by deeper, older forces, things like hunger, sex, maternal instincts, orchestrated by hormonal signals in one small structure of the brain? I'm talking about the hypothalamus, not just a part of the brain, but the heart of the brain, and the focus of a wonderful, eye-opening book called The Heart of the Brain, The Hypothalamus and Its Hormones by today's guest, Professor Gareth Leng. Welcome to the podcast, Gareth.
Thank you very much. It's a great pleasure to be here. And that was a lovely introduction, so thank you for that.
Thanks, Gareth. A professor emeritus at the University of Edinburgh, Gareth has had a long and distinguished career in neuroendocrinology, the study of how hormones and the brain interact. So he's more than well placed to guide us on a journey into this deep, dark recess of the brain and to shine a torch on a hidden command center that governs many of the behaviors we think of as deeply human. On this journey into one of the oldest parts of the brain, we'll be exploring how neurons in the hypothalamus orchestrate everything from mating, puberty, and maternal bonding to appetite, obesity, and circadian rhythms. So, if you've ever wondered what really drives your hunger, thirst, or libido, or why you wake up just before your alarm goes off, you've come to the right place. Gareth's path to science wasn't inevitable. He studied maths at the University of Warwick, but lost interest and applied for jobs as a teacher, a technical editor, and a systems analyst. And a careers advisor even suggested he try writing for Rolling Stone. And then something clicked. Inspired by a professor who believed maths could uncover simple rules behind complex behaviors, Gareth decided to give biology a try and never looked back. He retrained as a neuroscientist, earned a PhD, and was soon recruited by Barry Cross, a pioneering figure in hypothalamus research, at what is now the Babraham Institute in the UK. Barry gave him his own lab and tenure, despite the fact that Gareth did not yet know what a hormone was. As Gareth writes, "When I arrived at Babraham, Barry told me, 'Here's your lab, let me know when you found something interesting.'" As it turned out, that leap of faith paid off. But as you point out in your book, Gareth, opportunities like that are much rarer today.
That's right, and I think it's so sad. I wasn't unique by any means. I think all Barry Cross's recruitments were on the same principle. And what he chose to do is to try and be innovative. He thought he'd invest in young people without preconceptions and just trust them, give them the opportunity and trust them. And I think it worked. On a grand scale, it certainly worked for me. But I think you can look at all of Barry Cross's recruits and you can look at them now and think, it seems as though he didn't make a mistake. And I'm sure it is the fact that if you give young people trust and an opportunity, and let them explore their curiosity, foster their own ideas, then it will pay off big. And I think it did.
Yes, and it's now common for early career scientists to move from one short-term position to another, often for years before they're even considered for a lab of their own. So things have very much changed since those days.
Oh, it's clear. It's clearly a mad and stupid strategy that we've been following. There is a kind of schizophrenia about science funding. On the one hand, the funders say that we want innovation, we want new ideas, we want bold new discoveries, and we want to invest in the young. But in fact, all of their strategies are opposed to that. In fact, they reinforce conservatism. To be successful in the present climate, you've actually got to be productive, you've got to work in somebody else's lab, following their ideas, and producing papers along an established pattern. And where from there can you grow real innovation and real excitement? I think really the structure of science funding needs people to stand back and say, is it really what we want?
And to be less risk averse.
This is important. Funders say they want to fund risk, but actually what they do is they penalize failure. You can't have both of these things. If you really want to encourage people to try risky things, then you've got to understand that quite often risky things won't come off. So you've got to provide a kind of safety blanket. So you've got to trust people for the long term if you really want people to attempt novel and ambitious things and break into new areas.
So if you curl your tongue back as far as you can and press it on the roof of your mouth, the hypothalamus will be almost on the tip of your tongue. So that gives us a rough idea of where it is. But Gareth, maybe you can give us a quick overview of what it actually is and what it does.
Well, I suppose interest in the hypothalamus really exploded at about the time of my birth. It was really somebody called Geoffrey Harris, who was a scientist then, professor in Oxford, who wrote a monograph called The Neural Control of the Pituitary, which really set the agenda for what we now know as neuroendocrinology for the future. And so he's often now called the founder of neuroendocrinology. Now, his real contribution, his real breakthrough, was an outrageously extravagant hypothesis. What he did was he proposed that this tiny part of the brain, it still is a small part of the brain, which is actually just sits above the pituitary gland. Now, the pituitary gland is what's called the body's master gland. It produces a whole range of hormones that really control all of the other endocrine organs in the body and in many of our bodily functions. So it controls our ovaries or testes. It controls the thyroid gland. It controls the adrenal gland, which controls our stress responses. And so this master gland itself sits at the base of the brain. It's not part of the brain, but it produces these hormones that control the other endocrine systems. So in a sense, it's massively important for much of the things that go on in our body. And more functions of the pituitary are being found constantly. Now, Geoffrey Harris's contribution was that he said that, well, it can't be any coincidence that the pituitary is sitting below the brain. But we knew that, and he knew that there wasn't any direct connection between these two. The nerves didn't go into most of the pituitary gland. And so what he proposed is that, well, the neurons have to release something into the blood vessels that run from the brain into the pituitary gland. And they have to release messengers that control those. Now, this was a outrageously ambitious hypothesis at the time, because what he was doing was proposing this tiny part of the brain was making a number of different, unique, somehow special substances for which there was really no evidence at all, that each of which separately controlled a different hormone that was coming from the pituitary gland. And these were coming from neurons, and this isn't the kind of thing that neurons are supposed to do anyway. So it was an extraordinary hypothesis and a bold hypothesis. But there were people on the other side that were extravagantly opposed to it, outraged by this idea.
The idea that the hypothalamus produces signaling molecules, including hormones and peptides, that influence both the nervous system and the hormonal system? That was the idea that
Well,
was...
at the time, it was just the control of the pituitary. But this is where it came to in the end. The idea that neurons also produce hormones was really a shocking idea. But in fact, Harris's hypothesis was spectacularly validated because all of those releasing factors were in turn discovered, identified, and they won Nobel Prizes for people involved in doing it. It was a real breakthrough in endocrinology. And it shifted really, it put the hypothalamus really at the forefront of people's attention. But then people had already looked at the hypothalamus and knew that the hypothalamus was involved in all kinds of behaviors as well. It's a tiny part of the brain, as you've said, but we knew it was involved in fear responses, in aggression. Hints it was involved in temperature regulation. But there were also, even then, hints that it was involved in behavior, not just aggressive behavior, not just stress behavior, but also all the behaviors associated with reproduction. And as years went on, all of these behaviors came to the forefront as well. And the links between these behaviors and the control of hormones and the peptides that are controlling the release of the hormones became apparent. And it really became apparent then that these fundamental behaviors were regulated by, if you like, hormonal systems within the brain. These nerve cells were controlling not just the pituitary, but also other parts of the brain and controlling behaviors in conjunction with the release of the hormones.
And in the book, you compared the hypothalamus to the News of the World, that old British tabloid newspaper notorious for its sensational headlines, because, as the motto went, all human life is there. And in many ways, that's true. The hypothalamus is deeply involved in the raw, primal drives of appetite, sex, aggression, and more, as you were alluding to. And it doesn't stop there. When we think about the brain, we often focus on the cerebral cortex, the outer layer of the brain, and assume that because this region is more extensive in humans than other mammals, that is where our humanity and intelligence reside. But of course, many of our decisions, even the big ones like whom to choose as a life partner, are shaped or indeed subverted by an array of cognitive biases and emotional undercurrents. And here again, the hypothalamus plays a role, not just in our more base instincts, but also in some of our most sublime and emotionally rich experiences, such as maternal love, friendship, and bonding. And in the book, you explain that the cells of the hypothalamus are far from uniform. And in fact, they belong to many distinct subpopulations or clans, as you call them.
There was a time not very long ago when thought that neurons were just a single cell type. And that, yes, they had different inputs, but they were basically the same, and they worked in the same way, and they could talk to each other and be understood by each other. Now we recognize that there are, in fact, a vast number of different neuronal types. They're different in terms of the messengers that they make, in terms of where they project to, in the information they receive, in the way they convert the signals that they receive into patterns of electrical activity. I should explain that the classic way that neurons communicate is they generate electrical spikes that then travel down the nerve processes and cause the release of substances at their endings. And it's those substances released will affect other neurons down the road. So there is a link between the electrical activity that cells generate and the chemical signals that they produce. And it's the chemical signals that affect the other cells down the line. Now, if these cells are different in the way that they construct those electrical signals, then there's a problem because each cell has its unique language, if you like. And how do other neurons understand that language? And not only is that a problem, but now we know that most neurons don't make just one signaling molecule. They make several. And they release them according to different rules. And that is a shocking idea. It's a shocking idea because we've come to think that of the brain as though it's like a computer, that you have single track messages going down from one thing to another, carrying specific information. And now we see neurons communicating by a lot of different ways of talking to each other. They do talk neuron to neuron. They have a kind of single-track direct messenger system, which is called the neurotransmitters. But they also have a way of communicating to whole populations. They can release peptides, and peptides can diffuse and travel in different places, and they will find cells that have the receptors for those peptides. They're talking to different populations on different timescales and different spatial dimensions according to different rules. And so that is a challenge because we can't understand the way that the brain works without recognizing that neurons are very different from population to population and that they use multiple modes of communication.
Yes, and these spikes or action potentials, which you just described, they often hog the limelight. But peptides are a very important part of the communication equation.
You're right to say that spikes have hogged the limelight. I've been an electrophysiologist, by the way. I've been recording spikes for 40 years. I mean, that's what I did. That's what I came into neuroscience to do, and to understand how that spike activity, what information it carries and what it does. But really, this is a technique that we could do. Right? That's why it hogged it, because it became possible to do this. It became possible to look at this. We had the technology for doing it. When we discovered that actually it's not the whole story, that there's another mode of peptide release, that posed very different problems. To study that, you have to be able to measure a whole range of different molecules at phenomenal sensitivity from very small areas. And that is phenomenally difficult to do. So although we know that there's this peptide communication going on and we can see it happening in some ways, we can't see it happening in real time. We can't follow it in the same way that we can follow spike activity. Not yet. Many neuroscientists thought, okay, the hypothalamus is different and special, but we don't have to worry about it because we're not in the hypothalamus, we're interested in other areas. But actually they can't do that anymore because we now know that pretty well every neuron in the brain makes other messengers as well as neurotransmitters. This is a challenge that really no neuroscientist can escape.
So we have these glands in the hypothalamus, which send their own special mix of peptides, these large protein-like messengers, alongside the smaller, more familiar neurotransmitters involved in spikes like dopamine and serotonin. And you make a very nice analogy between whispers and public announcements. So while neurotransmitters involved in action potentials are like whispers, fleeting and localized, peptides, of which there are over 100 types, are more like public announcements, stronger, longer lasting, and broadcast more widely across the brain. They don't just influence signaling between neurons, they can also affect which genes those neurons switch on or off. And there's been a big push in neuroscience to understand the brain by mapping its connectome, the exact wiring between cells. But as you argue in the book, Gareth, to really understand behavior, it's not enough to know which neurons are connected. We also need to know where these chemical messages can land and where the receptors for these peptides and hormones are.
Yes, you're right. There's a lot of excitement about the development of our understanding of the connectome. And it's, just to give you a scale of it, what we're talking about in terms of the human brain, we're talking about a few billion neurons, each of which makes about 10,000 connections. So the connectome is a massive, massive network of neurons. And it's often been assumed that, well, it's so massive, then that's got to explain everything. I think that assumption is fundamentally wrong. I'm not saying that it's useless, it's clearly not. It's an ambitious aim. Will certainly tell us a lot that we don't already know, but it has its limitations. It will tell us which neurons are connected to which other neurons, but it won't tell us what signals are released at that site, what those signals do. It won't tell us how the neurons receiving those signals will interpret those signals, which will differ from cell to cell. And we shouldn't think that the connectome is universal and fixed. It varies from one person to another, one animal to another, and it varies with our experience and our development. But then even beyond that, we know that neurons use a great variety of messengers. We talked about peptides, but they also use gases, nitric oxide, possibly carbon monoxide as well. They use steroids. They make a class of things called neurosteroids, which are like steroid hormones. And they make prostaglandins. So they use a big variety of signaling messengers. So even if we understand fully the physical connections, we'll still only have quite a small part of the total way that neurons communicate.
You've already mentioned that brains aren't computers, and you make the case in the book that because the cells, neurons within our brain are messy and unreliable products of genetics, epigenetics, accident, and chance, this makes any analogy between computers and the brain misleading.
That's right. That's with the present generation of computers. I think that's true. But I think the interesting possibility comes with quantum computing. Our existing computers, which are digital computers, progressively for the last 40 years, they've been increasing in complexity and lowering in cost at a phenomenal rate, but a steady rate, an exponential rate. But there is a problem. The problem is that we're approaching what seems to be a kind of absolute hard limit on the way in which conventional computer elements can be miniaturized, where the gap between junctions is so small that actually you can't constrain an electrical signal to control its passage through. It will become random. And at that point, chip technology will become unreliable. And we're not far off that. So the computer industry is working frantically to develop a generation of computers that will escape this limit. And it can be escaped by what's called quantum computation. And quantum computation involves elements that really exist at the molecular level. Absolutely tiny, tiny elements. And now, if they can reduce the computing elements down to that size, then you have an infinite possibility of further miniaturization and further growth in power. But what we know is that if you're looking at computation at a molecular level, then what we know is that these quantum computation elements will firstly be intrinsically very noisy. They will be intrinsically very unreliable. In itself, that's not a problem because they can be so tiny, you can have masses, huge numbers of them. So you can require reliability through a massive number of these elements. The second thing, though, is that all conventional computing works on logic gates, a 0 or 1, true or false. Quantum elements have multiple states. So now that gives them massively more power, potentially, because you don't have to have a single element that makes a categorical decision. You can have every computational element responding to shades of grey. But the interesting thing to me is that if we think about how quantum computers, we expect them to work, they're actually quite close to the way we understand the brain actually works. They're closer to neurons, much closer to neurons than our conventional computers.
So computing as an analogy for how the brain works may actually be more accurate in the future when we have these quantum computers. You have talked about these clans in the hypothalamus and how neurons in each clan release their own special brew of peptides. And remarkably, when you inject these different peptides into the brain without any precise targeting or sophisticated delivery, they can trigger clear, meaningful behaviors. Some peptides can instantly spark eating. Others can create a craving for salt or sex, initiate maternal care, provoke aggression, or even induce sleep. And as you write, I find it hard to express just how shocking this is. It's as though you can take a sonnet of Shakespeare, cut out the words, and scatter them in the wind, only to find them gathered together faultlessly. Now, one of the key roles of the hypothalamus is to act as a kind of command center for the neuroendocrine system, as we've discussed, the system where the brain talks to the body through hormones. And we've talked about the pituitary gland, the master gland, which sits just below the hypothalamus and responds to its signals by releasing hormones into the bloodstream, helping to regulate everything from stress and growth to reproduction and metabolism. So Gareth, before we look at some of these examples, maybe you can paint for us in broad brushstrokes how these different clans in the hypothalamus control the release of hormones in the pituitary gland.
I call them clans. Living in Scotland, it seemed like a good name. But what I really wanted to do is to emphasize that actually these aren't clones. So the members of a clan are not identical to each other. They have family things in common. Everyone's an individual. Well, that's very much the same is true of neurons in the brain. So even though I talk about, for instance, the clan of oxytocin neurons. And there are a few thousand oxytocin neurons in a rat brain, maybe 20, 30,000 in a human brain. But each one is different. What makes them different is, well, these are, as you said, accidents of development. And they are the fact that no neuron gets exactly the same inputs. They all get slightly different inputs from different areas. Each one's got slightly different gene levels of gene expression, which gives it slightly different properties. We look at it and you think, oh, these are noisy, they're messy, they're all different. But actually, sometimes having a whole population of things that are very different gives you a lot more robustness and flexibility. So if they're all the same, they'd all have the same faults and the same weaknesses. What's interesting is that if you look at oxytocin, for instance, talked about Jeffrey Harris was talking about signals that could go into the blood and then reach the pituitary. Oxytocin is an exception. These are neurons in the brain that send their axons all the way down into the pituitary. So these neurons and this clan of oxytocin neurons in a small part of the hypothalamus produce hormones that go directly into the bloodstream. And what does it do in the periphery? Well, it depends on where the receptors are. So it gets into the blood, and to have any action, it's got to act on receptors. Now, one of the things that oxytocin does is it promotes labor in a pregnant woman at the end of term, helps with the delivery of the young. And it works at that time, because at that time, at the end of pregnancy, the uterus expresses huge numbers of oxytocin receptors. And it doesn't at any other time. So oxytocin, when it comes into the periphery, acts at the uterus, but it can only act at the uterus at the end of pregnancy when the uterus has got lots of receptors. And then after the baby is delivered, then the mammary glands are filled with oxytocin receptors, and now oxytocin is triggering milk letdown from the mammary glands. So what oxytocin does, even in the periphery, depends on the physiological state of the woman or the organism and on where the receptors are expressed in that particular physiological state. In rodents, for instance, oxytocin does something completely different as well. What it does in rodents is it controls sodium excretion through receptors in the heart and in the kidney. So the oxytocin then can do different things at different times depending on where the receptors are, depending on the state of the animal. The same is true within the brain. So oxytocin is released not only from the endings in the pituitary gland, it's also released within the brain, but it's released by a different mechanism in the brain. In the release from the pituitary is governed by these action potentials, these electrical signals that travel down the processes to the pituitary gland and trigger the release of vesicles that contain oxytocin. And in the brain, what we find is that there's lots of oxytocin vesicles, but they're present in what are called dendrites. These are part of a neuron, not the axons that go down to the pituitary. They're a different part of the neuron. And they're a part of the neuron we know receives inputs from other neurons. It's not conventionally the site at which there is neurotransmitter release. A dendrite doesn't make synapses with other neurons. But the dendrites of oxytocin neurons contain lots of oxytocin granules. Now, the signals that are received at the dendrites, some of those can cause calcium to be released from stores within the dendrite. And signals that can do that can cause oxytocin to be released from the dendrites. Now, it's a different signal because signals can cause intracellular calcium release even if they don't cause spikes to be generated. So that means is that depending on the signal that comes into the oxytocin neuron, you might get release within the brain or release within the pituitary, or both. Now that is shattering because now gave us a way of thinking that peptide secreting cells can do different things. They can do one thing at their endings, but they can do another thing by their peptide release at different times in response to different signals.
And oxytocin is perhaps the best known messenger in this system, sometimes called the love hormone, though that hardly does justice to everything it does. As you say, it's involved in a whole range of behavioral effects. As you've just described them, we have oxytocin release from two different parts of certain brain cells in the hypothalamus. One release point at the tips of their long fibers, which send oxytocin into the bloodstream via the pituitary gland. And this is the release that directly triggers the milk ejection reflex. And this is the one role where oxytocin is absolutely essential, because it's certainly important for things like mating and childbirth. You've mentioned labor, but it's essential in triggering the milk ejection reflex in breastfeeding mothers. And it does that through bursts of oxytocin release from the hypothalamus, without which milk simply doesn't flow. And so, if I'm right, Gareth, this oxytocin, it's not just released from the ends of the cells in the pituitary gland, it's also released from the branches of these brain cells within the brain itself. And these branches, dendrites, which is where cells receive incoming signals, the release of oxytocin from the dendrites is what acts on nearby oxytocin-producing cells in the brain, helping them all fire together in sync. And it's this coordinated burst of activity is what triggers the pulses of oxytocin into the bloodstream. And those pulses, not a steady flow, but pulses, are exactly what's needed to make the milk ejection reflex work properly. And bursts of oxytocin, I believe, are essential also during labor.
Yes, I think I can say that. During labour, it's a little bit more complicated because you've got an elevated background level as well as bursts. And exactly what the bursts are important for still isn't really quite clear. And that's because during parturition, the uterus is continually contracting in a kind of sinusoidal rhythm. And that those contractions are amplified when there's an elevated background level of oxytocin. So what do the pulses of oxytocin during labour do above that? It still isn't really clear. I don't want to give the audience the idea that we understand everything. We absolutely don't. It is possible that the pulses of oxytocin actually give the final large push that drives the baby out, but I think it's actually not quite that simple. I think in many ways it looks more as though when the baby is going through the cervix, it is that triggers the big pulse. So what is the big pulse doing? Is its importance triggering behaviour changes? So it is possible then that pulse of oxytocin that comes with the delivery of a baby, maybe its importance is in the effects on behaviour, the effects on the brain. We certainly know from studies in animals like rats that it seems that the vaginal delivery, the normal delivery, is important in animals for triggering the maternal behaviour that follows. If you deliver the young of a rat by caesarean section, then very often the maternal behaviour will fail. They won't nurse the young, and sometimes they will attack them. But if you give them oxytocin into the brain, then they will be normally maternal. So we know from experiments like that the oxytocin flow that goes into the brain at the time of parturition is important in animals for initiating that change in behaviour that is so important for the survival of the young.
You mentioned a couple of other fascinating studies showing the role of oxytocin in other behaviors. In one study, scientists discovered that they could make female rats behave like mothers, even if they've never had babies, by injecting a small amount of oxytocin into their brains. And this only works if the rat has high levels of estrogen. And in another study, mother mice naturally respond to the high-pitched distress calls of their pups. They'll go and retrieve them when they hear them. Virgin female mice usually don't do this, but if you inject oxytocin into their brain or activate oxytocin-producing brain cells, they start behaving like mothers, going to the pups and bringing them back. And it seems that oxytocin acts on the part of the brain here that processes sound, the auditory cortex, making the pup's cries stand out more. Even though only a few oxytocin signals reach that area, they're enough to create this lasting effect, helping the brain treat those cries as urgent and emotionally important. Now, one of the most famous pieces of research on oxytocin and bonding comes from a rather unexpected place: prairie voles. And these small rodents have become something of a neuroscience celebrity species because, unlike many other animals, they form strong monogamous pair bonds. And what researchers discovered in these voles gave us some of the earliest and clearest evidence that oxytocin isn't just about reproduction, it's also crucial for social attachment. Can you walk us through what those studies found briefly and why they were so important for our understanding of bonding and the brain?
Well, as you say, it's such a lovely story, a kind of unforgettable story, and that it resonates in so many different ways. The prairie vole became a very interesting model because its social behavior is very different to that of other voles, even voles that are living in the same part of the world in North America. And so the original studies were to look at what makes the prairie vole different from a related species called the montane vole. And let me just explain what the difference in behavior is. The prairie vole, the male, takes a full part in parenting. The prairie voles bond apparently for life. They're very sociable, very apparent really at first sight. If you put a male and female prairie vole in a cage, you'll find them cuddled up together. It's very visible. Now, montane voles are radically different. They are promiscuous. They don't form single bonds, and the male plays no part in bringing up the young. And you put two montane voles in a cage, they'll be at opposite corners of the cage. The difference is instantly visible. And so, of course, naturally you see this, and the question is, what makes them so different? One of the wild guesses is maybe it's the oxytocin system is different. And the oxytocin cells were the same in these voles as far as anybody could see. But the receptors are in different places. And that was striking. And they looked at a whole range of receptors, and it wasn't just the oxytocin receptors. The vasopressin receptors are also in different places. And so was this then the explanation for the different behaviors? Tom Insel and his collaborators made this model famous initially by his use of what's called a partner preference test. And this is a test to establish whether a male vole and female vole have actually got a bond. And the test is fairly simple. What you do is you take a female and give that female the choice of whether to spend time with either a male that she is mated with or a new male. Very simple. And for a prairie vole, there's no competition really. Virtually all the time, the female will go with and choose to spend time with the male that she's mated previously. Whereas the montane vole won't. Montane vole doesn't distinguish. It will just spend small amounts of time with either of them, enough to have a bit of sex perhaps, but that's it. So what does it take to establish a bond between prairie voles? Well, in the wild, essentially a prairie vole male meets a prairie vole female. They spend, if they can, a kind of weekend in intense sexual activity. More or less copulate continuously for about 48 hours. And it is this experience of intense proximity and sexual activity which seems to be enough to establish this long-lasting bond. So something happens during this time of intense sexual activity to cause that bond. And actually, well, that kind of fits because we've known for a long time that sexual activity itself will cause a large amounts of oxytocin release. So that's certainly consistent with the idea that oxytocin is triggering that bond. But it was Tom Insel really that led the experiments that really established that very clearly. And he did it in this way. He said that, well, if it's oxytocin that is responsible for this, then can we stop the bond being formed by giving an antagonist to oxytocin in the brain? The question is fairly simple. You introduce prairie vole male and female together. You allow them to have sex and interact in a normal way, but you give an oxytocin antagonist into the brain. Then you've got to ask, has a bond been formed? Well, now we come back to the partner preference test. So a week later, you take the female and give her the choice of the male that she's mated with or a strange male. And the fact is she doesn't seem to remember the male she's mated with, and she will choose at random. So it seems that you can block the formation of this bond using an oxytocin antagonist. Maybe even more strikingly, you can induce a false bond by giving oxytocin. So in other words, if you introduce a female to a male, you don't allow them to mate, but you let them interact through a mesh cage, and give the female oxytocin in the brain, and a week later, she will respond as though she's had sex. She will display a partner, a bond with the male that she was with at the time she had the oxytocin. So it did seem then that those experiments had established pretty clearly, pretty persuasively to most people in the field, I think at the time, that oxytocin released in the brain was instrumental in forming these bonds. Now, that's the female story, but we know that the males also form bonds. Males play a full part in parenting. And often the male bond is exhibited in aggression towards other males. The male is very kind of defensive of the female, protect its nest, protect the young, protect the female. So again, you can find tests for that formation of a bond in the males. Now, in the males, it's not oxytocin that induces the bond. It's actually vasopressin. And I mentioned that vasopressin receptors from different places as well. And the person really responsible for this fabulous story, which in many ways has become the leading story, was Larry Young, who was a great friend, was a wonderful scientist. Sadly, at a horribly young age, he died earlier this year. But I think his legacy in establishing the importance of vasopressin in bonding is going to last an enormously long time. It's a hugely influential story. Because he, of course, then took it a massive stage further. He wanted to know what is it about what makes the vasopressin receptors appear in different places. And he and his collaborators identified the differences in the vasopressin receptor gene that specifies where they're going to be expressed. And that now, with the current technology for generating transgenic animals, makes it possible even to develop mice, transgenic mice, who have vasopressin receptors in the brain in the same places that they do in the prairie vole. And what they've shown is that you can change the social behavior of mice by changing the distribution of vasopressin receptors. And changing nothing about the signaling from vasopressin, you're just putting the receptors in a different place. And that's enough to change the message that vasopressin carries.
So in prairie voles, in male prairie voles, vasopressin helps promote these bonding behaviors. But in the promiscuous montane voles, where those receptors are less densely expressed in key brain areas, it doesn't. And this goes back to what we were saying about the connectome and how it's not enough to know which neurons are connected. We also need to know where chemical signals can land and where their receptors are.
Exactly.
And remarkable that you can genetically modify mice, as you say, to carry the prairie vole version of the vasopressin receptor, and they start showing more affiliative, bonding-like behaviors. And it suggests that even small genetic differences in how these receptors are regulated can shape how social or attached an animal is. And we humans also have different versions of the genes for both vasopressin and oxytocin receptors. Is there any evidence that these variations could help explain why people differ in how strongly they form social bonds?
Yeah, it's a very interesting question. How important it is, I don't know. But it's a question that really kind of piques our curiosity, isn't it? And of course, again, this is an area that Larry Young explored. In other words, he went in to look for, are there associations between the vasopressin receptor in humans and their behavior? And the first thing that it became very clear is that in humans, the vasopressin receptor gene is massively variable between individuals. This is unusual in the genome, the human genome. Most of our genes are relatively consistent. They have limited variability between individuals, but the vasopressin receptor is an exception. There's a very wide range. So there are individuals with receptors that look more like the prairie voles, others with more like the montane voles. And so the only question you can ask, really, does that variability in any way kind of correlate with differences in social behavior? I think the jury has to be out on that. There have been some studies out there that suggest that maybe a relationship success might be associated with a different form of the vasopressin receptor. But to be honest, these studies are difficult to do, and their kind of conclusions are often dangerous to draw from inevitably small studies and complex studies, because relationship success depends upon so many things, not least it depends on two people, not one person. And there are so many other things in the background development that will affect this. So the question of whether the genetic makeup of the vasopressin receptors in humans has a big input on this is we really don't know. But maybe we shouldn't know. Maybe it's better we don't.
And interestingly, the hypothalamus does differ between males and females.
Yes, it certainly does. In a number of parts of the hypothalamus. Oxytocin cells and the vasopressin cells that project to the pituitary, they don't differ. The main difference in the hypothalamus is in groups of cells that express receptors for estrogen. But it's not just those, it's also cells, different populations of cells have receptors for testosterone. And you find the receptors in both males and females for estrogen and testosterone, but they're in different places and in different amounts in different parts of the hypothalamus. So there are in the hypothalamus parts that we regard as sexually dimorphic. And in fact, there are populations of vasopressin cells in the hypothalamus that are sexually dimorphic. And these are populations that express receptors for estrogen.
So far, we've seen that how a hormone is released, its timing and pattern can be just as important as which hormone is being released. And that's exactly the case in the example we're about to look at. Now, there are two key hormones made by the pituitary gland, luteinizing hormone, or LH, and follicle stimulating hormone, or FSH, which control everything from sperm production and ovulation to sexual development and puberty. But their release depends on another hormone called gonadotropin releasing hormone, or GnRH, which is made in the hypothalamus. What's fascinating is that GnRH isn't released in a steady flow. Yes, it's sent out in pulses. And these pulses travel through a special set of blood vessels to the pituitary gland, which then responds by releasing LH and FSH into the body. So, Gareth, can you explain why that's so important for triggering ovulation and the release of an egg from the ovary?
I will just give you a glimpse of the answer. And I just want to talk about one of the hormones, which is LH, luteinizing hormone, which is released from the pituitary gland. And you're quite right, the what we call the ovarian cycle, or the menstrual cycle in females, is really governed by a changing pattern of release of luteinizing hormone. At all times in the cycle, luteinizing hormone, if I can call it LH, if I may,
Yes.
LH is released in pulses. But the size of those pulses and the frequency of those pulses varies progressively through the course of the cycle. In a rat, the cycle is all over in 4 or 5 days. In a human, it's 28 days. But whether you're talking about a long cycle in a human or a short cycle as in a rat, the same things are generally true. What you've got is a progressively changing pattern of LH secretion. Now, this pattern of LH secretion, as the amplitude increases, then that drives the development of the ovary. But actually, to get that final process of ovulation, you've got to have more than these pulses. You've got to have something called a surge. So the pulses in the end culminate in a massive release of LH. And this massive release of LH causes the event that we call ovulation. So the pulses through the cycle help the egg mature. And once it's matured and is ready, you need a big pulse of LH and out it pops. So the whole cycle is really governed by the pattern. It's governed by the changing pulsatile pattern and by the emergence of this LH surge. And as you said, all of this is in turn governed by the secretion of a releasing hormone. We tend to call it GnRH for gonadotropin releasing hormone because the pulses of GnRH.
The GnRH from the hypothalamus.
So the GnRH is released from hypothalamic neurons, and it triggers a pulse of LH release from the pituitary. If you try and give LH continuously, then you'll desensitize the receptors. And if you try and give GnRH continually, you'll desensitize the LH secreting endocrine cells. So you have to have a pulsatile signal to stop this desensitization. It makes the signal more efficient. you'll get the result by using less of the hormone, whether it's GnRH or LH. So you need the pulses. And then at some point you've got to change, you've got to produce this big surge. And that's been a problem because it seemed that the surge is controlled in one part of the hypothalamus, but the pulses are controlled in a different one. So how can that be? It's a real kind of mystery. And the second thing was, obviously, if you've got pulses, the cells have got to be synchronized. So actually, the cells have got to be pulsing together. So they should be talking to each other. But when people found where the GnRH cells are in the hypothalamus, they found that they weren't gathered together in one convenient place. They're all over the damn place. They're really scattered. Now, of course, the cells are scattered all over the place, but their axons come together at the median eminence where the endings release their substances into the vessels that reach the pituitary gland. Themselves are further away up in a different part of the hypothalamus, quite a distance away. So, solving that problem has been one of the great achievements of neuroendocrinology over the last 10 or 20 years. And the solution to that problem came with the discovery of another peptide, a new peptide that's called Kisspeptin.
Which has nothing to do with kissing, by the way. There's not
Has
some kind of
absolutely
romantic
nothing. It's
molecule.
actually named after Hershey's kisses. The gene was supposedly discovered by a group working in Hershey. The Kisspeptin cells appear to be controlling the GnRH cells, but the Kisspeptin cells are close to the terminals. The cell bodies of the GnRH cells are a long way away. But then there was another population of Kisspeptin cells that was close to the cell bodies. So now we had two populations of Kisspeptin cells, one close to the terminals and one close to the cell bodies. And the next step was to show that the Kisspeptin cells actually are sensitive to estrogen levels. Now, the GnRH cells aren't. They don't have any estrogen receptors. And so the Kisspeptin cells seem to be the ones that respond to the signals that are coming back from the ovary to the brain.
They act as a kind of messenger, telling the GnRH neurons when to fire.
That's right. We're ready for a surge now, so come and give us a surge, please. Now, what's interesting is that the cells, the Kisspeptin cells in these two locations, respond in a different way to estrogen. They respond in opposite ways. And now that becomes important because actually the surge occurs at a time when the pulses are being inhibited and are slowed down. And so you can now see that you've got one population of Kisspeptin cells that activates the GnRH cells to produce the surge. The other populations seem to be important for the pulses. But the other population is down by the nerve endings. It's not the place where you should be controlling the electrical activity. And we've talked about how important spikes are. Spikes are normally generated at the cell body. This is the site at which this signal is controlled. So what on earth is going on? How can cells down at the terminals be controlling the pulsatile release? Well, that involved a radical rethinking of how neurons are constructed and what the GnRH neuron is really like. And Alan Herbison, who was one of the leaders in driving this understanding, recognized that actually we need a new vocabulary. What we thought of as an axon coming from the cell body down to the median eminence, where the blood vessels are that reach the pituitary, that process actually doesn't really behave like a conventional axon at all. It behaves in part more like a dendrite. So he called it a dendron. What it means is that the Kisspeptin cells that act close to the terminals can activate release from those terminals. And the Kisspeptin cells that are close to the cell bodies can change the electrical activity of the cell. So you've got two different kinds of independent signals. So it looks as though the Kisspeptin cells close to the terminals can drive a pulsatile release of GnRH by activating the end of the dendron. But that's quite a different message is involved in the generation of the surge from a different population of the Kisspeptin cells acting at the cell bodies of the GnRH cells, not the terminals. So this is a kind of really a revolution in our understanding of this neuron. It's groundbreaking in so many ways, but it resolved all of these problems that had been apparent, these confusions and these anomalies, present in our understanding for 30, 40 years or longer.
It's an amazingly intricate system. And an important point you make, Gareth, is that the hypothalamus here doesn't act alone and also needs input from the ovaries. Because just before ovulation, the estrogen levels from the ovaries rise sharply, and this stimulates a surge of GnRH release from the hypothalamus, which in turn causes a surge in LH from the pituitary gland. And it's this LH surge that triggers ovulation. So it's a good example of how, even though we often talk about the brain controlling reproduction, in reality, the brain, pituitary gland, and ovaries are all in constant communication, with each influencing the other. And so it's hard to say that any one single part is really in charge. And these Kisspeptin neurons are the ones regulating the GnRH and ensuring that we have this burst of GnRH, because they are sensitive to estrogen, whereas GnRH neurons are not. Have I summed that up?
That's absolutely right. That's absolutely right. Yeah, neuroscientists, I would like to think that the brain's in charge of everything. But actually, increasingly, we're realizing that signals from the periphery and hormonal signals have got huge effects on our behavior.
And since we're talking about the reproductive cycle, I'll just bring in a quick example from one of our own labs at the Champalimaud Foundation. A recent study led by Susanna Lima's team uncovered a specific brain circuit in female mice that helps control sexual rejection. They found that a group of neurons in the hypothalamus becomes active when a female isn't fertile during the cycle, prompting behaviors like kicking or avoiding the male. And when the female is fertile, those neurons quiet down, allowing mating to happen. And it's a great example of how the brain can flip behavioral responses depending on internal state. The same stimulus, in this case a male mouse, can trigger completely opposite behaviors depending on the phase of the cycle. And it really highlights just how flexible and dynamic these hypothalamic circuits are. And what do we know about the clinical impact of this hormone system, the GnRH, LH? Has our knowledge of how this system works helped in tackling infertility and precocious puberty?
Certainly in terms of infertility, in terms of in vitro fertilization, for instance, when our understanding of this is actually absolutely critical to the preparation of a woman for in vitro fertilization. There are a number of kind of contraceptives, long-lasting contraceptives that have been introduced actually using agonists of LH or agonists of GnRH, and that they work on the basis of this desensitization that I talked about. You need a pulsatile signal to avoid desensitization. So if you give a long-lasting agonist continuously, you will desensitize the tissue and effectively get a contraceptive.
And these GnRH neurons in the hypothalamus are pretty extraordinary, because not only are there very few of them, they actually start life not in the brain, but in the nose, and then migrate into the brain. And there was even a paper in the journal Nature in 2013 that suggested that the decline of GnRH contributes directly to the process of aging, and that lifespan itself can be extended by maintaining this population of GnRH neurons. And speaking of lifespans, the lifespan of this episode has come to an end. Despite the hypothalamus's small size, we simply couldn't squeeze it all into one episode. But fear not, our journey into this deep, dark cave of the brain is far from over. In the next episode, Gareth returns to shine his torch even deeper into the hypothalamus. And it gets even weirder. You'll hear about the experiment where researchers transplanted brain tissue from one animal to another, resetting the host's internal clock, and meet a molecule so strange it can reduce appetite, boost libido, and even give you a tan. We'll also look at obesity and explore how the brain balances body weight and how vasopressin doesn't just shape behavior, but helps keep you alive. So, whether you listen to this podcast in one continuous sitting or in pulses, stay tuned for our next episode and see you then.